So we're going to begin our Regents review with a discussion of scientific inquiry, and then we're going to move on to homeostasis. So I'll be going through this presentation relatively quickly because we've already talked about all of this, but just gives us a chance for me to emphasize certain points about each of the topics for the Regents exam. So when we talk about science, science is a process of inquiring about the world, of making hypotheses, testing those hypotheses in a way that's repeatable by other people to double check or to support those experimental results. So we start with observations about the world around us and then ask questions. And once we have those questions formulated and we have something we're trying to figure out, science starts with or continues on to form a hypothesis. Hypothesis is a statement, a prediction about what is going to happen in the experiment. And that hypothesis is tested through experimental design. So an experiment is set up that's going to test the prediction that's made by the hypothesis, and that hypothesis can be supported by the results of the experiment. It might not be supported, but this informs our understanding of the problem. So when we talk about experiments, what we are referring to in science are what we call controlled experiments. Controlled experiments attempt to test how one variable, one factor, is affecting the results. And so when we talk about these controlled experiments, we have variables which have specific names that you should certainly understand and be able to differentiate between. So in an experiment, the dependent variable is the variable, I would say, that we're sort of interested in the result of. The dependent variable is measured. I often say it's the data we're collecting. To help you remember, the D in dependent goes with the data that's being collected. And the independent variable is sort of the factor, the thing you're altering, the thing that you're changing to see how it affects the dependent variable. The independent variable in a good experiment, there should just be one. You want to try to keep as many factors the same and only alter the independent variable. And if you're able to do that successfully, when you change just the independent variable, you look at the dependent variable to see how that's been affected. When we write a hypothesis, there's some important points to remember. It has to be a statement. A hypothesis can never be a question. Often a hypothesis is phrased as an if-then statement. If I do this to the independent variable, then this will happen to the dependent variable. Let's look at the example on the right side of this slide. So if we're thinking about scientific inquiry, we start with an observation that both poisonous coral snakes and non-poisonous king snakes have the same color rings. That's an observation. So we may ask a question, well, what's the function of the king snake having the same colors as a coral snake? That's a question we're asking. So we can come up with a hypothesis. The hypothesis might be that this mimicry of the coral snake helps the king snake avoid predators. That's a hypothesis that we can test in an experiment. So I make a prediction. If predators confuse king snakes with coral snakes, then the predators should attack fewer ringed artificial snakes than brown artificial snakes. So you could set up an experiment. You could have these artificial snakes with different colorations, and you could measure the number of attacks on these artificial snakes, and that would allow us to test that hypothesis. So in this experiment, let's talk about the variables. The independent variable are the different types of snakes, the ring snakes versus the brown snakes. The dependent variable would be the number of attacks on each of those snakes. Variables I would want to control for might be the age of the snake, the sex of the snake, the type of enclosure that they're in, All of those things should be kept the same so that I can be sure that only my independent variable is different in my experimental setup. And so when we observe nature, this often leads to questions, and those questions can often lead to hypotheses, which can be tested in an experiment. And that's done in collecting data. Okay, so data is the measured results of our experiment. So if I'm looking at this chart, we have age and we have height. Age is in years, height is in centimeters. And so in this case, the independent variable would be the age, and then the dependent variable would be the height. And I'm measuring the height of people at different ages. So when we design a controlled experiment, let's say for this example, we're designing an experiment that's going to test how effective a specific cough drop is. We need to start with stating our hypothesis. Okay? If I'm doing that in this experiment, my hypothesis might be that this cough drop will reduce the number of coughs per minute in individuals that have a cold. Okay, so the independent variable in that experiment would be the type of cough drop. Maybe some individuals in the experiment would get the cough drop, others would not. I try to keep the other factors the same. They should be the same age. They should have the same type of cold. The dependent variable would be the number of times the person's coughing. This is the data I'm collecting that's going to help me make my conclusion. The control group would be the group that's not getting any cough drop. I would collect my data, how many times each person in each group coughs over a period of time, make a data table, probably average the results to see the overall number of coughs per minute. And then if I get results that confirm my hypothesis, I might repeat it again. I do another experiment to be sure that this experiment correctly gives the same information. If I want to design an experiment to test if a new fertilizer increases the growth in plants, I might have 10 plants in each group. One group is getting this new fertilizer, the other group is not getting the new fertilizer, just getting water. My hypothesis might be this new fertilizer will help the plants to grow taller. In that experiment, the independent variable, the thing that's different, is the fertilizer or not fertilizer. All other factors would be the same. They're getting the same amount of water, the same amount of light, they're the same type of seeds. The dependent variable, the data I'm collecting, would be the height of the plants. The control group would be the group that's not getting any fertilizer. It's just getting regular water. And all those other factors would be the same. I'd be measuring all of these plants, every day for a period of, say, 3 weeks. And then I would look at my data in a graph, likely the average growth of the fertilized group over a period of time graphed, and also the average growth of the non-fertilized over a period of time, and look at the differences between those and potentially repeat the experiment. So that's scientific inquiry. Definitely you should know the different types of variables, the control group, the experimental group. Often there will be questions when they describe an experiment and then ask you to analyze it and say, what is the dependent variable? What's the independent variable? What is the control group, etc.? Or there may be questions in which they propose a hypothesis and you have to describe an experiment, a properly controlled experiment that meets the requirements that we've just talked about in this video.